All right. Hi, everybody. I hope that you are all doing well. Couple of general things to talk about before I get started. First of all, if you are following along with the stuff on YouTube, I posted the 2 lectures from this week already. I have not yet done last week's because those are the ones I have to re-record. But since my recordings are working fine this week, I got those done. So at least get some things off my plate. So those are there, and the ones from last week will be showing up this weekend. I also spent a little bit of time looking at the schedule that is upcoming. And so I made a couple of changes. There is an updated schedule posted on Moodle. So I want to tell you a little bit about what I did. I don't love it. And you'll see why I don't love it as I'm talking about it. So what we're going to be doing next week is that basically I've taken the paper that we were going to discuss and moved it later. So it is not next week. Instead, we're talking about generation of antibody diversity on both Monday and Wednesday, and then immunology techniques on Friday. The trick is that the generation of antibody diversity and the techniques both involve problem sets. And I want you to have time after you learn the stuff to actually come talk to me about it. But I also want to have time to actually comment on your problem sets before the exam. And so those 2 problem sets are due the Monday and Wednesday the following week. They're already posted. You could start working on them now. And in fact, we're going to do the paper discussion on Friday of that week. And so there's discussion papers for that Friday. So there's basically something due, not next week, but the week after, all 3 days. Hate it. But it's the only way I could sort of pull things together before the exam. All of those assignments, all of that paper that we're discussing, all of that is posted. So even though it's not for more than a week, it's there for you to start on because I feel so bad about the way that week got loaded up. So be aware of it now, and know that the point was that you could come by and ask questions and talk to me about stuff for office hours. And all of the things are posted. I'm always happy to answer questions about any of that stuff. So today we're going to be talking about aspects of antibody diversity. And there are lots of different directions that one could take the idea of antibody diversity into. But I think that all the topics that I'm talking about today can sort of go under that broad umbrella. So if you recall, last time I introduced you to this idea of antibody isotypes. So we have our 5 different antibody isotypes that vary based on which heavy chain constant region they are using. Yes, light chains also have a constant region. Nobody cares. We'll care for 2 minutes in a couple weeks, but no one really cares. And you can see that those are the mu constant region, which makes IgM, D, which makes delta, which makes IgD, gamma, which makes IgG, epsilon, which makes IgE, and alpha, which makes IgA. And if you recall, last time we talked about the different effector functions of antibodies, so the different functions that these antibodies all have. What we need to spend some time talking about today are these 5 different isotypes and what's actually unique about them. So that's how this falls into antibody diversity. It's the way that these 5 different isotypes are different from one another. I've already showed you the tables that are on the next 2 slides at the very end last time, and you're going to see them a bunch more throughout today. So don't feel like you have to know the whole table when I show it to you right now. I'm going to point different things out throughout the day today. And so what we're going to be thinking about really are sort of these unique aspects of our different isotypes. This does actually show you that we have multiple types of G, multiple types of A. I'm going to I will mention a couple little things about the different subtypes of G, but right for the most part, we're just going to lump them all together. And you can see that things like the size of the antibody, so the molecular mass can vary, the serum level, so how much is floating in the blood in the serum, and the half-life in serum, or how long those antibodies last in serum, are all unique to some of these different isotypes. And you can also see that there are some functions and properties that differ between the different isotypes. So as we go through each isotype, these are some of the types of things we're going to talk about. I told you last time that this order, MDGEA, is also important for some things. I'm not going to tell you about them in this order, however. I got reasons, but we're just going to go with it. Although I am going to start with IgM. And one thing to sort of think about with IgM and the fact that I'm starting with IgM is that immune responses also start with IgM. So when you are first making a new antibody to something, it's an IgM antibody. When your B cell starts making antibodies, it makes IgMs. So IgMs are always made first, and I'm going to tell you about IgM first. So if we look at this table, I've put a red box around IgM. What is kind of the thing about IgM that is a standout here? Yeah, Brooke. Yeah, IgM sort of is a standout because of this large molecular mass. And so one of the things that is special about IgM has to do with the molecular mass. And what's going on with that very large molecular mass with IgM is that the IgM antibody is not generally found in your body as an individual antibody molecule. Instead, IgM is generally found as a pentamer. So you have 5 IgM antibodies hooked together with something called a J chain. And so IgM really looks like this as pentameric IgM in your body. Pretty much as soon as it's made, it ends up going into this pentamer form. And you can actually, again, see some IgM pentamers under electron microscopes here at the bottom. And so this is why IgM has such a large molecular mass. Why might you imagine that this pentameric structure, or what kind of bonus or benefit do you think this pentameric structure gives IgM? It gives it one big bonus that we need to think about. Yep, Nadia. Oh.
It's okay.
It's okay.
I was thinking of an answer.
Okay.
Can it attach to more things? Because there's more.
Yeah, it can attach to 10 copies of the epitope instead of just 2. So why do we care that it can attach to 10 copies of the epitope instead of just 2? If you're unsure about this, imagine what it would be like if I have my cat who I really like at home and I had 10 arms instead of 2 arms. What would the key difference be? Yeah, Jazz.
Basically, like just a few more in terms of like, especially for antibodies, like it's able to attach to more epitopes, and for things that have multiple epitopes on a single thing, it just attach to multiple of them.
And so because it can attach to multiple of them, what ends up happening? Or what's the result? You know this, you're just like thinking past it. I really love this cat. This is my cat. She's not actually here. She would hate being here. So no, not just engulfing it, but you can grab it. You can grab it with more force. You have a stronger ability to interact. You have a higher ability to bind. If you're just grabbing it with 2 things, it's weaker than if you're grabbing with 10, right? And so IgM has 10 binding sites, so it can really bind on quite well to our antigen. This actually becomes important later because, as we'll eventually see, the antigen binding site in IgM is, the individual antigen binding site is kind of weak, but it doesn't matter because it's got 10. So it's got 10 and they're all next to each other, so you get really strong binding even though the individual binding sites are actually a little on the weak side. Yep.
Is the molecular mass so high because it's all 5 of them?
Yeah, because it's all 5 of them plus this J chain
Plus the J chain.
that's holding it together.
What was that again?
So this J chain, which you see here, is the thing that's actually holding the 5 together.
Oh, okay. All right.
Yeah. So this pentameric structure is one of the big things that's really unique about IgM. I've also mentioned IgM being sort of first made. That is pretty important, and that's something that will come up later on. But this structure of IgM also leads to the other really unique thing about IgM. And so if we look at this table, again, I've put a red box around IgM. So what is unique about IgM, or what do you see here as being really key for IgM? Yeah, Jazz. IgM is really good at turning on complement. This shows IgG kind of doing it, but honestly, if I think about complement, I'm thinking about IgM. IgM is the complement all-star, particularly the classical pathway complement all-star. So remember, the classical pathway was the one that was started by an antibody. The antibody that is by far the best at starting the classical pathway is IgM. What was the first step in the classical pathway after the antibody bound to the antigen? What happens next in the classical pathway? Yeah, David. So now C1 is going to bind to the antibody, right? And C1, in fact, binds, if I'm being now I can use this term, it binds to the Fc portion of the antibody. I couldn't tell you that before because I hadn't defined Fc portions yet. So it binds to the Fc portion of the antibody. What does a C1 look like? Do you remember? It has a very specific structure. Yeah, Ani. Looks like an upside down bouquet of flowers. When IgM is in its pentamer form, it's in this form with the 5 Fc tails together. It fits the upside down bouquet of flowers perfectly. It's like the exact perfect geometry match to C1. And that's when it's in that form with the 5, when it's in the pentamer form. And so you can see this is why IgM is so good at starting the classical pathway of complement. Other antibody types can do it if they sort of luck into being in the right geometry. But IgM, because of its 5 tails being held in a specific way, is always in the perfect geometry to start the classical pathway. And so that's why IgM is really the all-star in terms of starting the classical pathway of complement, because it just looks, it puts the Fc portions in exactly the right place for C1. So I'm going to switch gears. So other immunologists would be like, how did she just go from the first one, IgM, to IgD, or to IgE? Like, what is she doing? This is not the right order. But the order makes sense. I have a reason. So you can look at IgE here, and you can see some information about IgE. And so my question for you about IgE is, where is IgE unique? What's different about IgE compared to the others? Yep, Paige. Its serum level is really, really small. IgE has the lowest serum level by far. And you can also see It's tied for lowest in terms of half-life. So the idea here is there's not a lot of IgE in the serum. You might look at that and say, oh, perhaps that means we don't make very much IgE. Or you might look at that and say, is the IgE somewhere else in the body other than the serum? Because this is telling me the serum level, not the level in the whole body. And it turns out the answer is that the IgE is somewhere else. The IgE is not in the serum. So you have lots of IgE, it's just not floating around in the serum. And so this location issue is a key thing about IgE. IgE has the epsilon constant region, right? As a result, IgE can bind to Fc epsilon receptors, the Fc receptor that binds to the epsilon constant region. And as soon as IgE is made, it sticks to any Fc receptor in the body. So basically, your IgE is all stuck on cells on their Fc epsilon receptors. It's not floating around free in the blood. So it's all over your body, stuck on any cell that has an Fc receptor, not floating around by itself. That unique feature of IgE also kind of tells us about ends up leading to its unique function. Because when I just told you about IgE, I said it's on Fc epsilon receptors on cells all over your body. But I didn't tell you what cells they were. I just said cells. It turns out there's really only one kind of cell in your body that has the Fc epsilon receptor. So basically, as soon as your IgE is made, it coats that one cell. It gets stuck to the Fc epsilon receptors on that one cell type. And so you can see IgE, we're going to ignore this box right here. IgE really has one function, and it's pretty much the only antibody that has that function, the only S type that has that function. So what is unique about IgE? Yep, Jazz.
Sensitization of mast cells.
Sensitization of mast cells. So IgE sensitizes mast cells. And you can see it's really the only one that sensitizes mast cells. IgE has this pretty tight linkage with mast cells. And so basically, all of your IgE is bound to Fc receptors on your mast cell. So all of your mast cells are like pre-coated with IgE right now. And that's fine. Your mast cells are just floating around with IgE on their surface. But if the antigen that IgE binds to is ever present, that will bring together these Fc receptors, and it will make the mast cell release. So basically, the mast cells are always like super on the trigger, ready to go, because they already have the IgE all over their surface. They're already sitting there waiting. And if the antigen ever comes around, they will activate and they will release the materials in their granules right away. And so this is sort of that effector function of activation of mast cells, which is only done by IgE. Paige, you had a question?
So the only the big difference between IgE and all the other antibodies is because they don't have to go through the step of the antibody finding the receptor because it's already there.
So sure. It just never hangs out free in your blood.
Okay.
It just gets pre-stuck to the mast cells. It goes in your blood and immediately it binds to the first mast cell it finds. And it's just sitting there on the mast cell ready to go and making the mast cell ready to go. Yep, Jazz.
So, if IgE is always ready to rumble, how come IgM is made first in terms of immune response?
So there's a lot that can be said in the answer. Some of answering of that is later genetics. But one thing I will tell you is that some of this also has to do with how useful some of these different functions are. So you can imagine turning on the classical pathway of complement. That's pretty useful. You kind of want to have that happening pretty early and pretty much right away. I haven't told you a ton about the function of mast cells yet. I've told you that they release this stuff in their granules, but I haven't told you really what the stuff is. I've kind of implied that it's toxic, but that's about all I've said. That stuff in the mast cell granules is there are a lot of different toxic materials. The most famous thing in a mast cell granule is histamine. And these are things that can cause a lot of damage to a lot of cells. They are really good for attacking extracellular parasites or things that are way too big to phagocytose. You can't phagocytose it, so instead you shoot toxic bombs at it. And so this is really good for parasite infection, but if you have mast cells triggering against other things, like, I don't know, in my lungs when they trigger against pollen, then you have some serious issues. And so one thing that's also sort of unique or interesting is that we do have some control over when we make IgE, which we don't have in the same way over when we're making IgM. That's my simplified answer that I could go on and on for a long time about. So that's IgE. I feel like IgE is pretty straightforward. So now I'm going to go back to sort of standard immunologist order of M, D, G, E, A. So I did M, so now I got to do D. And on the next slide, I'm showing you another one of those tables like I've been showing you before. But I'm showing you a table from a slightly older textbook. And so this is an earlier edition of a textbook, but it's the same kind of table. And I've got the red box around IgD. And so I'm going to show you this table, and I want you to tell me, according to this table, which, like I said, is a little bit more out of date. Oh no, I didn't make the more out of date one. Okay, there's an even more out of date one. I swear I thought I had the super out of date one on here. If you look at a really out of date textbook, the answer to what IgD does, the function of IgD, is nothing. It has no known function. And so there would be a minus sign everywhere for IgD. And when I started teaching at Drew, I taught that IgD had no function. It's just like a weird evolutionary relic. And I said the only thing that anyone cares about of IgD is that it is on the membrane of B cells at a particular point in their development. So sometimes we use it to help us figure out where B cells are in development. There's a certain place where they have IgD on their surface. So it's like a nice marker for us, but biologically no one cares, is the way that we used to talk about IgD. And so here you can see we've got all these different stages of development of the B cell, and at one particular point it makes IgD on its surface. And so I learned IgD simply as a thing that helps you know where a B cell is in its life. In other ways, I didn't care about it. Since I've been teaching at Drew, the story on IgD has changed a little bit. And now IgD has one function. And so if you look at this table, what's the function of IgD according to the table? Yep, Abigail. So this is showing IgD as sensitizing basophils. The basic idea with IgD and sensitizing basophils is that it looks really similar to IgE on mast cells. So it's not necessarily that the IgD is maybe pre-bound, we think. We think that the mast cell or the basophil releases granules at the same way the mast cell does. We think the IgD basophil thing is really similar to what I just showed you with IgE and mast cells. I don't even have a picture of it in a slide because this is so relatively new and people don't draw anything about, don't have pictures of it. The issue here is that if you recall back to when I told you about cells of the immune system and I mentioned basophils, I told you they are the most rare cells in the blood. You have like no basophils in your blood. As a result, nobody studies them because it's really hard to get them and do experiments on them. So there's very little that's known about basophils. And so things like maybe IgD and basophils are doing something super important and we just can't discover it because there's too few of them to actually look at. So it's only really recently we realized IgD is doing something with basophils. There's still a lot that's not known. Former member of my lab's doing lots of cool basophil stuff and he gets mad at me that I don't talk about how great they are. So if you want to know more about them, tell me and I'll get you in touch with them. I'm sure he's listening to this recording, so hi. So the final two things, so I've told you about IgM, I've told you about IgD, I've told you about IgE. I now have to talk about IgA and IgG. And I'm going to start with IgA. So if you look at this table, yeah, you can see there's two kinds of IgA. I wouldn't get too stressed about that. Is there anything that really stands out to you about IgA on this table? IgA is pretty average in all of these like types of things. So there's nothing like super exciting about IgA here. But IgA does have some really key functions. So if you look, you can see that IgA is really good at neutralization. The thing that's really special about IgA, that makes IgA so unique compared to other isotypes, is actually where in your body the IgA is found. So again, IgA has some really unique anatomical locations, and that's what's interesting about IgA, more so than any other particular aspect of its function. It does all the functions like decently well, neutralizes very well. It's just it does them in a unique part of your body is really what's key about IgA. So oftentimes IgA, in fact, is found as a dimer. It also has its own J chain holding it together to have two copies of it. This dimerized IgA is known as secretory IgA. When I think about IgA, I think about secretory IgA in general. I have a friend who is a co-host of one of my podcasts who works on IgA, and she sometimes gets feisty and she's like, it's not all secretory. There's some that's not a dimer. So Steph says there's some that's not a dimer, but I'm only going to really talk about the dimer versions, though if you want to know more about that, we can read stuff from Steph. And you can see some examples of that secretory IgA here. And IgA gets into a unique part of the body through a process called transcytosis. And so if you imagine different parts of your body, you might imagine, say, your intestine, right? This is a very, very detailed drawing I'm going to make. So there's the inside of the intestine where the food goes, okay? We've got sort of a layer of intestine cells around that area. So here's my intestine cells. And you can kind of see that we have this like cavity in between our intestine cells, which is where the food goes. And then we've got, some connective tissue. Here's some tissue. Here's some tissue, right? And somewhere in all of this, there is also a blood vessel or some blood. And so maybe my squiggly can be the blood. Okay? So we've got some blood and some vessels in the tissue. Then we've got this like wall of cells. And then we have this open cavity, right? And you could imagine the same kind of setup in your lungs. The part where the air goes, a wall of cells, the rest of your body with the blood. You can imagine this in your nose. You can imagine this in your mouth. You can imagine this in a lot of different parts of your body where you have mucosal areas. The thing that's interesting about IgA is able to cross this wall of cells and be in that empty cavity. And so you don't necessarily have any immune cells in there protecting you where the food is, but you've got a bunch of IgA antibody. And so if, say, you eat a pathogen, it can get neutralized in your GI tract before it starts to impact the rest of the body. So the key thing about IgA is it's in this mucosal area because it's able to be carried across the cell wall, the cell layer, the wall o cells. It's not a cell wall, it's a wall o cells, to get into that mucosal cavity. And so here you can see IgA, it actually is like bound to a specific receptor in that wall of cells, carried in a little compartment across, and then put into the cavity area. And this is called transcytosis. And IgA is the only isotype that can undergo transcytosis. So in your intestine, you just have IgA. In your mouth, in your saliva, you've got a bunch of IgA. In your reproductive tract, you got a bunch of IgA. In your eyes, you got a bunch of IgA. Basically, IgA is sort of like the thing that's outside the border, trying to neutralize a microbe before it gets in. And it's able to cross that cell border by this process of transcytosis. Here you can see this sort of general process. So here's what is this supposed to be? This is supposed to be a lung. So here's the area with the air, and here's the cell layer, and here you can see some bacteria in the lung, and they might reproduce there, and you might get sick, and that would be sad. But happily, we have some IgA in this location, and that IgA can neutralize the bacteria before they even are able to sort of really establish themselves in the body. So they're sort of like, so IgA is the isotype that's sort of outside the gate in your mucosal area. Yep, Jazz.
So, mucus in regards to like as like in our nose or whatever.
Mm-hmm.
In terms of physically, it's like sticky, so that's what makes it innate. But then, because it has IgA within it, it makes it adaptive as well.
So there are more things than just the stickiness that make mucus an innate response. But it does, in fact, have some, I would say, adaptive immune molecules. In fact, because of this nice wall we have here, you can even call it barrier. Or because it has mucus, you can even call it barrier. So it's actually, again, you can, what you've highlighted in your question is the way that these layers work together and sort of blend together. Yep, Paige.
Is IgA always found in the inside, or if there is a pathogen that's...
No, you always, right now, if I wanted to get some IgA from you, I could have you spit in a tube. You always have IgA in all of your mucosal sites kind of ready to go. So IgA is always being made and going to mucosal sites to protect you there. Steph would tell me that there's also some that's not in mucosal sites that's in the blood, and it's important too, but I'm not going to talk about it. There's one other really interesting detail, or actually there are two other interesting details about IgA. One of them is also related to anatomic location. Because we've talked about the intestine, we've talked about the lung, we've talked about saliva, all sorts of places where IgA is found, but there's one other location where IgA is found. And so you can see you can see this person's body. It color coded based on which antibodies are in which place, and you can see that the yellow is all the mucosal sites, and that's only IgA. So IgA is the only one that gets to those mucosal sites. Other ones are in the blood in other places. I don't know why they drew IgE the way they did, but I'm going to ignore that for right now. But IgA also is in breast milk. And so this ability to do transcytosis also means that IgA can be in breast milk. And so when a mom breastfeeds, she actually gives her baby IgA, and the baby's intestine gets mom's IgA before the baby's able to make their own IgA. It kind of makes sense because baby's probably going to get infected with the same kind of things mom might have been infected with in her life. So giving her some of mom's antibodies is useful. And so that's the other key thing. And in fact, babies who are breastfed are protected by their mom's IgA for the amount of time that they're breastfeeding. So they're kind of getting this boost to their immune system. Yep, Emily.
So
No other antibodies are able to cross. No other antibodies are able to cross into breast milk. We will see the baby is protected with another antibody in another way, but it's not from breast milk. So only IgA in breast milk. Yep, Jazz.
I don't mean to ask a lot of questions.
That's fine.
But,
Go for it.
How does that work with babies who are formula fed instead? Is it supplemented or like fortified through their?
It is not. So yes, there is no IgA in formula. There are a bunch of things that we now know are sort of different between what's in formula versus what's in breast milk that we're only in the past 10-ish years learning. And so it's possible that in the future we could design a new formula that has some of the benefits we now know of in breast milk. But yeah, we don't have any, the formula does not have IgA supplemented in. One thing I will also point out, this comes up more later today, I think. I'm now like, okay, yeah, it comes, I'll talk about it later. I know why. I know why I don't have it here. I figured it out. There's one other thing that's really interesting about IgA, which is that having a deficiency where you don't make IgA is actually the most common immunodeficiency. And so this is actually showing a table from your textbook of the number of people who are IgA deficient per million in different groups. So you can see that it's found in different frequencies in different groups. But it's still pretty common. It would not be like surprising for one of you to be sitting here and be IgA deficient and have no IgA. Most of the studies that are out there on selective IgA deficiency say that people who have it are completely healthy and it's totally fine. And so that might be part of the reason why people are like, why put it in the formula? You can just not have it at all and be fine. That might be because of modern hygiene, and maybe in the past when hygiene wasn't as good, this may have been more of a problem. When people start to do really sort of detailed epidemiological studies, there are some questions about whether people who are IgA deficient may have more chronic lung disease if they live in an industrialized country, perhaps because of air quality and air pollution. There's some evidence there may be some relationship with celiac disease, but none of it is like super strong evidence. You really need a lot to sort of figure out what's going on here. And one thing that sometimes people will think about with this is, well, okay, so it's super common. Why didn't we have evolutionary selection against this? Sometimes people will say, well, if it can be so common that you don't have IgA, must not be that important. If like evolution didn't kill all the people who didn't have IgA really quick, must not be a big deal. But one of the things that we think about is that what isn't something that is likely to be eliminated by natural selection, because babies who don't make IgA still have IgA. They just have their mom's IgA, right? And they're going to be particularly at risk of microbial infection. And so even if you don't make IgA as a baby, so you'd be really, really at risk of dying of some microbe, you get it from your mom, especially in past human times. And so it's hard for natural selection to kind of act on that deficiency in quite the same way that we might normally think of. And so that may be why this actually has not been selected against and is still present in the human population. Yep.
Do you keep your mom's IgA your entire lifespan?
Nope, you just keep it for maybe depends on how long you're breastfed.
Okay.
But you only keep it basically, yeah. But you're also at most risk when you're really young. So it's sort of like, yeah. And so that's the other kind of interesting fact about IgA. The other thing, so the final isotype that we need to talk about is IgG. You can see that there are actually 4 different subtypes of IgG shown here. And if you got into a more advanced immunology textbook, it would actually divide them up more. So there's a bunch of different types of IgG. They are relatively average in a lot of things. What sort of stands out for you about IgG? And I might especially say, look at something like G1. IgG1 as being the standout. Yeah, Abigail. Long half-life. So it's not, especially IgG1, it's not getting degraded very quickly. It's sticking around for a long time. What else? There's one other thing that's sort of unique, especially when you look at IgG1. Paige. High serum level. You got a lot of it. This is the one you have the most of. So if we were going to take the 5 and rank them, IgG is the one you definitely have the most of. It's by far the most common. One of the reasons why it's the most common is because it sticks around for a long time and it has a long half-life. You can see if we look at our different types of IgG, they're all really good at neutralization, and they can also they're pretty much the only ones that do NK cell activation. They can do some opsonization, they can do some complement. So they can do a lot of functions. My brief mention of the different IgG subtypes, just so you know, is here. You can see the different types of IgG. And what do you notice in the picture is different about them? What's the actual difference between IgG subtypes? Yeah, Paige? The hinge region. So you can see big differences in the hinge region. And particularly the length of the hinge region. It turns out that length of the hinge region is directly related to the half-life, how long the antibody sticks around. IgG3, which has the crazy long hinge, also has the crazy short half-life because it can get cut up easier. Proteases can attack it easier because it has an easier place to attack. That's sort of as much as I wanted to mention about what the differences are. I'm for now going to lump them all as IgG. And I've told you that there are a bunch of things that IgG is pretty good at a lot of functions. And to me, one of the most important things about IgG is you have a lot of it. So if you take antibodies from a person, most likely you're going to have IgG. IgG is kind of the one that's all around. A lot of people when they think of antibodies, they just think of IgG because there's just so much of it compared to all of the others. The other thing to sort of point out here is I've sort of implied this a little bit already, but we make different isotypes at different times in an immune response. So in black, you see IgM production. The first time that you are seeing some kind of microbe, whether it's vaccination or infection. You see IgM, and you see IgM's kind of happening early. I've told you before, IgM happens first. And then in red, you see IgG. And you see IgG takes longer. IgG happens later. But when it finally gets going, it's more. And so if you look in a patient and you see they're making IgG, you know they've kind of made their ways into their immune response. And if it's their second response, if they're making a secondary response, it's largely an IgG response. Yeah, there is a little M, but it's really all G. And so a lot of times what we might do is, if we're measuring whether or not someone is making an antibody response to something, we'll look, do you have any IgG? That might mean that you have memory response to it. You've really kind of made a robust response. And so IgG is often used as a way to indicate making a secondary response. In reality, you could use IgG, IgA, or IgE because of the timing that they're made. So you could use G, A, or E for this. But there's like no E in the blood. The A is not that much in the blood, it's mostly mucosal sites, whereas G is like, there's tons of it and it's really easy to measure. So part of the reason is just because there's so much IgG, it's really easy to measure. So that's the one you have the most of when you're making a big adaptive response to something, and so it's the one that people tend to measure the most frequently. There is one other unique feature about IgG. IgG also has one other kind of unique thing with regard to anatomy. And so IgG is unique because IgG can actually go across the cell layer that makes up the placenta. And so IgG can go from a mom to a fetus. And so you can see that a fetus, when it is in mom, only has IgG, that it came from the mom, that mom gave it. It hasn't necessarily made its own antibodies yet. So basically mom's giving the baby IgGs, and if she breastfeeds, she's also giving the baby some IgAs. And IgG is able to cross the placenta through a process that's pretty similar to the transcytosis process that we saw before, where it actually goes from, there's a wall between mom's blood and baby's blood. We don't want mom's blood and baby's blood to mix. So there's a placental wall between the two of them. And IgG can actually get transported across that wall from mom's blood straight into baby's blood. And so we get IgG into baby circulation. Just so you are, just because this becomes interesting, this is sort of the information about antibodies in infants. And so you can see here's birth. At birth, baby really just has IgG from mom. In these other colors, these are the baby making its own antibodies. And you can see at birth, it's maybe a little bit, but not too much. And that baby really doesn't start making much in the way of antibodies till about 6 to 9 months. So sort of at this maybe birth to 6 months period, the baby's pretty vulnerable, right? The baby's immune system's not really able to sort of do much on its own here. The baby's at big high risk. And then the baby's immune system's finally kind of matured enough to the point where we can make this work. So this is interesting in a lot of ways. People who have issues with vaccines sometimes say one of their problems is that there are too many vaccines given at times that they don't think should be given to babies. This is why they're given at that time. Basically, we pick 6 months because it's the first time the baby's immune system can actually use the vaccine. It's the first time the baby's immune system's ready to go and ready to make an immune response. If you go earlier, the baby's not going to do anything. The baby's immune response isn't going to do anything. And you might even have some of mom's leftover antibodies to mess things up. At about 6 months, however, the baby's super vulnerable and ready to respond. If you wait longer, you're basically making the baby be vulnerable longer. And so the idea is that we've used these information to say, aha, right here is where we want to protect you ASAP. And that's why the vaccine schedule is set that way. There's a very good reason that vaccines are in fact given right at that point. And so when people are sort of being feisty about it, I can go on and on about vaccines for a very long time, and I certainly will. There is no risk of having a bunch of vaccines at 6 months. The six month thing was actually very carefully chosen. Yep, Juliana.
I get the part about like at 6 months is when they can like start synthesizing. So I don't know which ones exactly or how many, but they do get some before. So how does that work with the immune system? Like, how does it work in their body?
So so they're making an immune response, but it's not as ideal as it could be. And most of those are things where the risk is so great that even having the sort of meh immune response is better than nothing. Okay. Does that make sense? Yeah. One other reason why this is sort of interesting is, as you can see, mom is giving a bunch of antibodies to babies. And so also in the time that I've worked at Drew, the vaccine recommendations from the CDC have changed. We'll even imagine before 2024 that they changed while I worked at Drew, before 2025. And so actually it is now a big thing to give a lot of vaccines to pregnant moms, so that those pregnant moms make tons of antibodies and give them to the baby, so that the baby can get the best possible protection. In fact, the most important one, the one that the CDC has really done a lot of influence with, is the pertussis vaccine. So pertussis is lethal in newborns. It's awful. I do not recommend any kind of Googling of a baby of a videos with baby with pertussis, because they're so sad, where these babies can't breathe and are just coughing, and it's awful. And it really is going to kill that baby before you can get any immune response going in that baby. And so what they actually do now is they give pregnant women a pertussis vaccine in their 3rd trimester, so that they make a lot of antibodies and they pass them all to the baby through the placenta and through breastfeeding, so that the baby can be as protected as possible. And there are a number of different models where people are spending more time looking at how can you vaccinate pregnant women so that you can in fact improve child health and improve newborn health by actually using this placental transfer, and by using this breast milk transfer. And again, I actually have a couple of friends who this is their whole life's work, is working on this stuff. It's really cool. Yeah.
I know lots of moms in their trimester intentionally pump to stock up on colostrum. How does this kind of tie into that?
So pretty different. It's not really related. So it would be a question of, I guess, where is your antibody boost related to when you're doing that, and the things that people are caring about in the colostrum is not so much the antibodies. Yes.
So this idea of giving the moms the vaccines, how would that affect them? If it's just a lot of stuff, is it kind of just it would just go somewhere?
No, it's fine. So here's my slight vaccine discussion. Right now I'm standing underneath an air vent, and there are microbes blowing on me from the air. I am seeing more antigens right now that are coming to me from this air vent than you ever do, even if you got every vaccine together on the same day. The amount of antigens you see from all those vaccines together is way less than just what you encounter in daily life. So the idea of getting too many at once is not a thing. I actually don't understand why CVS and Walgreens both have rules about how many vaccines they'll give you in one day. I have maxed out both of them at different times. And I don't know why they have those rules, honestly. There's no good immunological reason. So sometimes people are like, no, I don't, it helps protect mom, but it also helps protect baby. In fact, they often now a lot of pertussis vaccine is sort of being given out in a lot of places, because what they really want to do is cocoon infants. They want everybody around an infant to be vaccinated and to be protected, so that infant does not get pertussis vaccine. And so in fact, at one point when my mom went to her doctor's office, her doctor's like, well, you're at the age where a lot of women become grandmothers, so I really think we should give you a pertussis vaccine to protect your grandchildren. And then I got an angry phone call. So there's sort of a lot that's being done to really make sure that infants cannot get pertussis, this being one part of that. Yeah, Nadia?
Sorry, but
Yeah.
so then wouldn't that kind of work for every vaccine that mom kind of had already? Like the antibodies would just go, like, because I know a lot of newborns get hepatitis B.
Yeah.
Like that's like controversial or whatever, but if mom has it, then wouldn't they get it anyway, kind of?
No. So the hepatitis B thing is actually related to if you end up with hepatitis B at birth, it's super dangerous.
Yeah.
And so the hepatitis B vaccine they give at birth actually starts your protection immediately to keep you away from that danger. It's actually the infant.
Okay.
The hepatitis B vaccine for in a newborn is like crazy important for that newborn's health.
Yeah.
But this isn't the one I wanted. Hold on. The one I wanted is this one. So yes, every vaccine mom had is going to transfer to the baby. But as is kind of shown here, if I were going to draw this plot, when I draw this plot out later in the semester, you will recognize it. I would draw this plot looking more like this. You do see some decline over time, it's just slow. And so basically if mom is here when she's pregnant, she only has a little bit of antibodies to give to the baby. But if you give her the vaccine and she's here when she's pregnant, she's giving the baby a lot of good antibodies. And so that's why you're gonna vaccinate mom sort of right at they sort of have figured out timing of when do you vaccinate mom during pregnancy to make sure that she's given huge amounts of really good antibodies to that baby. One other piece that is a little bit relevant, or that is just sort of related and interesting, is that there are some autoimmune diseases that happen because you have antibodies that are a problem. So for example, there are some autoimmune diseases where you have antibodies against your thyroid that give you a disease called, it's called Graves' disease. There are women who have Graves' disease. This is clearly not to scale. Here's the thyroid. Here's an antibody. Here's mom having eye symptoms. These bad antibodies go across the placenta just like any good ones do. And so, in fact, that baby would have those antibodies and could potentially have some issues. We have ways to treat that. It's not like a thing, like we can prevent this now. So there are ways to deal with this. But note that any antibody, whether it's bad or good, would cross the placenta. If it was an IgG antibody. And so, yeah, we have ways to deal with this now. Also realize that in this situation, this is called passive antibody transfer. I told you about passive transfer of antibodies when I told you about von Behring and Kitasato, where the baby's just getting antibodies from mom. And this is true whether we're talking about the placenta or the breastfeeding. Baby's just getting antibodies from mom. AKA, she's just getting those proteins. She's not getting the baby's not getting the cells that make the proteins. The baby's just getting some of the proteins. The baby doesn't have any cells that made those proteins. And so the proteins are going to degrade at some point. The baby's not going to have these bad antibodies for life. The baby's going to have them for a few months until those proteins degrade, and then the baby's going to be healthy. And in fact, we have other ways we treat it now, so they're going to be healthy even before that. But so know that when you do passive transfer of antibodies, whether it's like I showed you before for treating a black widow spider bite, or whether we're thinking about antibodies from mom going into the baby, maybe even from that pertussis vaccine, those antibodies are only going to be in the baby for a short period of time, not a crazy long period of time, because it's just the protein that's being transferred across the placenta or through breastfeeding. It's not the actual B cell that makes the antibody. So once the proteins have degraded, which they degrade over time, then those antibodies are no longer going to be present. The baby needs to then make their own with their own B cells. So I titled this lecture Antibody Diversity, and now we're going to get into the other side of antibody diversity, what is sometimes referred to as the antibody diversity problem. And this is a really key problem in immunology. Again, this is one of those places where, yes, you might see slightly different numbers on different slides, and the point of the exact number doesn't matter. The thing that matters is the range of the numbers and sort of the comparison between the numbers I'm going to show you. So I've told you before that when we try to estimate or sort of come up with how many different antibodies there are, how many epitopes we can recognize in terms of our antibodies, we have this number that is about 10 to the 16th. Okay, yeah? So now I'm going to ask you a couple questions that we can pretend Dr. Dunaway is here and that you have the answers from Dr. Dunaway. So these are things you should have learned in Bio 250. Okay, be ready. Where does a protein come from? Because these are proteins, 10 to the 16th different proteins. Where does a protein come from? Yeah? The ribosome, okay? Ribosomes do translation of proteins. How did this ribosome know what to make? From an mRNA. So you had to have an mRNA, right? So you might imagine there had to be 10 to the 16th mRNAs. Yeah? Where does an mRNA come from? DNA, right? It's transcribed from a gene, right? So if you just think about it like this, you're like, okay, so we got 10 to the 16 different genes, make 10 to the 16 different mRNAs, make 10 to the 16 different proteins. Yeah? That's a problem. And the people who actually were thinking about this problem that I'm going to tell you, they figured out this problem in like 1900. The numbers, they didn't have 10 to the 16th as their number. They had a different number. But the problem was still the same problem. Even however accurate your numbers are, you get the same problem. So that's why I don't care what numbers we're actually using. We now know how many genes are in the human genome. Oh, and let me just... There's my 10 to the 16. Okay? We now know how many genes are in the human genome. And it's 2.3 times 10 to the fourth. $23,000 genes. So we got a problem here. How the heck are we getting that many antibody proteins out of that many genes in the genome? And guess what? Not only do you have that many antibody proteins, you have that many T cell receptors too. And we still don't have enough genes. And even I, as an immunologist who thinks about this stuff, am pretty sure that you can't use all your genes for antibodies and T cell receptors. You also need like insulin and actin and all that stuff. So there is a big math problem here of how the heck can you have this many proteins coming from that number of genes? There were, as I said, a bunch of scientists who tried to come up with different hypotheses about this. When they were making their hypotheses, some of their hypotheses are super weird. Sometimes I go through them and make fun of the scientists who made them. Just so you know, all of these scientists, if I do the making fun of them thing, all of the people I make fun of won a Nobel Prize for something else. So it's not like it's going to be a real problem for them that I make fun of them. But some of them ideas were super weird because they were trying to take a bunch of different observations that had been made and try to make a model that made sense of all those observations. And so I want to tell you what the observations are that our model has to be able to answer. So one of the things that our model has to be able to answer is that we have to have our B cells making this B cell receptor and the antibody, and they're basically the same. So all of our B cells are making the same thing, either with a transmembrane domain to be a B cell receptor or without one to be an antibody, to be a secreted protein. So that was one thing people were trying to figure out what the deal was. They also knew this. I'm going to define this term. I'm going to tell you what's going on here. It's going to sound wacky. We're going to see this term again in like a week, and it's going to be much more important and make much more sense then. But this is a phenomenon called allelic exclusion. And the idea with allelic exclusion is that each B cell makes a homogeneous set of receptors. What that means is another way of phrasing that is each B cell makes one receptor. It's not, you don't have one kind of B cell that makes all 10 to the 16th different proteins. You got 10 to the 16th B cells, each one makes one kind of antibody protein. So no B cell is making like 2 of the 10 to the 16th. Every B cell makes one and only one. It might make many copies of the one, but it's making only one unique antibody protein. And the other thing that they were trying to explain is that they knew that B cells and antibodies were part of this secondary or adaptive response. And so as time went on, we would get an increase in antibodies. And the second time, we would actually get even a bigger increase. So somehow, this response had to involve like being able to remember what you made previously and make more of it. At one point, there was a person who said that, for example, that B cells basically had this thing on their cell surface that was a blob, and whenever an antigen bound to it, the blob would shape around it. And then once that happened, eventually the blob would fall off, and that would be the antibody. There's 2 big problems with that. But by the way, the person who came up with this was the person who got the Nobel Prize for discovering blood groups. So again, we're not feeling too bad for him. 2 problems with this. First of all, for any of you who have thought about biochemistry, how do you feel about this blob that folds around an antigen business? The answer is no, that's not how biochemistry works. No, 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 that's not how biochemistry works. Second of all, and so the idea that they came up with was, his idea was like, well, each time the antigen binds, it folds a little bit better and it improves over time. That's how we get this improvement, and it's so great. But the other problem is a problem so you've got this amazing cell that undergoes this all these changes and builds this perfect antibody. What happens if that cell is in your finger when you get a cut on your finger and you bleed it out? It also is pretty dependent on that cell not getting lost or nothing bad happening to that cell. It's pretty dependent on that one cell. Because that one cell who changed its protein blob can't give its new protein blob change to its progeny cells, can it? No, we don't inherit acquired characteristics. So if we want to be able to give this really cool, interesting receptor to the progeny cell, what has to be involved? Yeah, Paige. Okay, but what if it's going to be transmitted to another cell. What's the main thing that's transmitted to the other cell? The number one most important thing that goes to the next cell from mother cell to daughter cell. Yeah, Elizabeth. DNA. The DNA's got to be involved somehow. You can't just have the proteins doing all sorts of funky things. Somehow we got to involve the DNA if we're going to actually have this be able to move from cell to cell. And one of the things that we sort of now know that's really important for this process is we now know that this process that I'm going to tell you about of this diversification that B cells do to make antibodies, it's the same process that other lymphocytes like T cells do to make their diverse receptors, is going to be a process that's going to involve DNA and is going to involve the cell making lots of different receptors. So we're going to have a stem cell, it's going to make lots of progeny cells, and each progeny cell is going to make a unique receptor. And so we've got this sort of process of making super diverse receptors through some stuff going on with the DNA. This is happening in the primary lymphoid organ before the cells go out and see pathogens. So first, they just diversify. Then they go out into the rest of the body and hopefully meet up with their microbe of interest, hopefully for them, I guess. And then they divide and divide and divide and make a clone army. Their clone expands. And we get more of them. Hopefully for you, they don't see their antigen and you live happily ever after. And so what we've realized is that what has to be happening is there has to be something interesting going on with DNA here early before we even see the microbes. When we're developing our lymphocyte, we've got to have some important stuff going on with DNA. So I'm going to mention this slide. And I may not start with this slide on Monday. It's going to be interesting. This slide is a great slide and also is a little bit problematic because you're going to love this slide when you're studying for the exam because this is like an outline of what I'm going to tell you next. So we have this problem, how do we make 10 to the 16th antibody proteins when our genome only has 2.3 times 10 to the 4th genes? There are two kinds of solutions. One is called combinatorial diversity, and there are two parts of combinatorial diversity. One is called junctional diversity. Next week on Monday and Wednesday, we're going to talk through what all these words mean that I have not defined yet here. So first we're going to think about combinatorial diversity and then see how combinatorial diversity works. Then we'll add in some junctional diversity. So I may not start with this. Next time I may just start into combinatorial diversity, but this is a nice outline or overview of what the answer is to how this happens, so that when you're studying later, you can see what these parts are. I will see you guys on Monday. Remember that not next week, but the week after, there's a bunch of stuff going on in this class, and so you might want to be thinking about some of that. Please let me know if you have questions.